Have you ever thought about what it would take to rewrite the very code of life itself? Well, today we're diving into a technology that does exactly that. It's called recombinant DNA, and it's a revolutionary process that lets scientists literally take a piece of genetic code from one organism and pop it into another, completely changing what it can do. So let's get into it. Just think about the possibilities for a second. What if you could turn a common bacterium, something like E. coli, into a tiny, living factory that just churns out life-saving human insulin? Or what if you could engineer a plant to grow a vaccine? This isn't some far-off sci-fi dream. This is the reality made possible by recombinant DNA. So at its heart, recombinant DNA technology is really just a super sophisticated form of genetic cut and paste. You're literally taking DNA from two different species, joining them together, and creating this brand new hybrid, sometimes called a chimeric, DNA molecule. The whole point is to install a new piece of code that gives an organism a new, powerful ability it never had before. All right, so to really get how this all works, we're going to break it down into 4 parts. First, we'll look at the molecular toolkit you need. Then we'll walk through the genetic assembly line where the magic happens. After that, we'll figure out how scientists find the one successful clone out of millions. And finally, we'll explore the absolutely revolutionary impact this has had on our world. Okay, first up, the toolkit. The best way to think about this is to picture a genetic engineer's lab as this high-tech inventor's workshop. But instead of wrenches and screwdrivers, the tools are microscopic proteins called enzymes, and each one is designed to do a very, very specific job when you're rewriting the code of life. These tools pretty much fall into 4 main categories. You've got your cutters, the nucleases, which act like molecular scissors. Then you have your pasters, the ligases, which are like molecular glue. You've even got enzymes that work like a photocopier, the polymerases, making millions of copies of a gene. And finally, you have others that make tiny chemical edits. Altogether, these give scientists an incredible amount of control over DNA. Now, the first and maybe most important tool in the whole kit is the restriction enzyme. These are the molecular scissors. And what's so cool is that bacteria actually evolved these as a defense mechanism to chop up the DNA of viruses that try to infect them. Scientists realized how incredibly precise they are. They only cut DNA at very specific sequences. And often, they make these staggered cuts that create what we call sticky ends. These little overhangs are absolutely crucial because they want to pair up with a matching sticky end, which makes them perfect for pasting a new piece of DNA right in. Okay, so you've used your scissors, you've made the cuts, you've opened up a space for your new gene. Well, now you need glue. That's where DNA ligase comes in. This enzyme's whole job is to repair breaks in the DNA backbone. It swoops in and permanently pastes our new gene fragment into its new home. It's the final step that seals the deal. So you have your engineered gene, awesome. But how do you get it inside a living cell? You need a delivery truck. In genetics, we call this a vector. And the most common vectors by far are plasmids. These are just small circular pieces of DNA that are naturally found in bacteria. They can copy themselves totally independently of the main chromosome, which makes them the perfect vehicle for carrying our new genetic instructions into the host. Right, we've got our tools: scissors, glue, and a delivery truck. Now let's head over to the genetic assembly line and see how they all work together to build a brand new molecule from scratch. The process itself follows a really clear logic. First, you isolate the specific gene you want to clone. Then, and this is key, you use the exact same restriction enzyme to cut both your gene and your plasmid vector. This is how you get those matching sticky ends. Next, you mix them together and add the DNA ligase to glue them up. Then comes a step called transformation, where you basically coax the host cells into taking up your new plasmid. And finally, the really critical step, screening. You have to find the cells that actually worked. And this diagram gives you a great visual of the process. You see the plasmid vector being cut open. Then the target DNA, the piece you actually care about, gets inserted. The DNA ligase comes in and seals it all up, and boom, you have your final recombined molecule. This new, souped-up plasmid is now ready to go into a host cell. But here's the kicker. That transformation step, it's not very efficient at all. You can mix millions of bacteria with millions of your new plasmids, but only a tiny fraction of them will actually take one inside. As you can see here, most of the potential host cells just fail. So how on earth do you find the successful ones in that massive crowd? This brings us to a huge quality control problem. You've done all this careful work to build your recombinant DNA, but now you need a way to sift through millions, maybe billions of cells to find that tiny fraction that has exactly what you're looking for. So yeah, that's the real challenge, right? Your petri dish is a total mess,
Most of the cells didn't take up any plasmid at all. Some took up a plasmid that just closed back up on itself without your new gene, and only a tiny, tiny fraction have the actual recombinant plasmid you spent all that time building. How do you find that needle in a haystack?
Well, the solution is nothing short of genius. It's a technique called insertional inactivation. Here's the idea. Scientists design the plasmid so that the spot where you insert your new gene is right smack in the middle of another functional gene. So when your gene gets inserted successfully, it breaks that original gene, turning it off. It's a brilliant trick. Your success is actually marked by breaking something. Let's look at a classic real-world example to make this super clear. We'll use a famous plasmid called PBR322. This plasmid naturally has two genes for antibiotic resistance, one for a drug called ampicillin and one for tetracycline. Now, the spot where we cut it to insert our new gene is located right inside the tetracycline resistance gene. So, if we successfully stick our new gene in there, we break the tetracycline resistance. But the ampicillin resistance gene is untouched and still works perfectly fine. And this gives us a foolproof way to find our successful clones. Think about it. A cell with the original unchanged plasmid is resistant to both ampicillin and tetracycline. But a cell with our new recombinant plasmid is different. It's still resistant to ampicillin, but because we broke the tetracycline resistance gene, it's now sensitive to tetracycline. So we just look for the cells that can grow on ampicillin but die when we add tetracycline. Those are our winners. All of this incredibly clever work in the lab leads to one big question. So what? What can we actually do with this? Well, in this last section, we're going to see how this technology jumped from the lab bench and went on to completely rewrite huge parts of our world. The impact has been absolutely staggering. We can now produce life-saving medicines like insulin for diabetics and growth hormone for people who need it, and we can do it on a massive scale. We can develop safer, more targeted vaccines. We're even creating gene therapies to treat devastating genetic disorders. And in agriculture, we can design crops that resist pests, survive droughts, and produce more food. It has touched almost every part of modern life. This technology basically gave birth to a whole industry of what we call biopharmaceuticals. A lot of modern drugs, especially really complex proteins like antibodies, are just impossible to make with normal chemistry. So instead, we use recombinant DNA to turn living cells into those tiny factories we talked about, and they produce these powerful drugs for us. And get this, for an application in vaccines, scientists can take a totally safe virus, like the one used for the smallpox vaccine, and insert genes from a really dangerous pathogen, like the surface proteins from malaria, influenza, or even HIV. The result is a vaccine that can train our immune system to recognize the bad guy, but without ever exposing us to the actual dangerous virus. It's a much safer and unbelievably versatile way to make vaccines. And this tech isn't just limited to microbes. There's a whole field called molecular farming, where scientists are turning entire plants into bioreactors. We can engineer a tobacco plant to produce human growth hormone, or get corn to make industrial enzymes. We can even get plants to produce the proteins for unbelievably strong materials like spider silk. It's wild. So we've seen the tools, we've walked through the process, and we've seen the incredible power of recombinant DNA. It has already rewritten medicine, agriculture, and our basic understanding of how life works. But the thing is, this technology is still young. The power to directly edit the blueprint of pretty much any living organism is now in our hands. And that leaves us with a profound and pretty exciting question. What will we build next?